Alright guys, so today we are going to discuss the mode of action of enzymes. Now there are two models presented by different scientists. One of the models is called as lock and key model, and second one is called as induced fit model.
So this is mod or model?
Model.
The
This
heading.
is mode. Mode means how do they work. First of all, we are going to talk about the lock and key model. Now, you guys have learned about the lock and key model in O levels. Now, in the lock and key model, the concept is that as there is a specific key for every lock, there is a specific enzyme for every substrate, right? And in this lock and key model, the substrate is equivalent to the key and enzyme is equivalent to the lock. The question is, why is it like this? Because a key fits into the lock. A key fits into the lock. Same, the substrate fits into active site of enzyme. As a key fits into the lock, the same way the substrate fits into the active site of the enzyme. Now, according to this model, the shape of substrate is an exact match to the active site of enzyme. The shape is exact match of the substrate to the active site of the enzyme. Now, for understanding that, let's suppose that here we have an enzyme. Let's suppose that this is the enzyme. And this is its active site. Now, enzymes and the substrate, they are present in the cytoplasm, right? Now, let's suppose that in the cytoplasm there are different types of substrates. There is one substrate which is like this, another substrate which is like this, another substrate which is like this. Now, which substrate is perfect to be fit into the active site? This one. Right? So, this substrate is an exact fit to the shape of the active site. What will happen? That this substrate, it will bind to the enzyme's active site. We discussed it yesterday as well, that this is the enzyme's active site, and over here, let me show it with the other color. The substrate fit into the active site. Now, when the substrate is fit into the active site, this is called as enzyme substrate complex. Now, we discussed this yesterday, that this enzyme substrate complex formed is for a very limited or short period of time. What will happen? Right away, this enzyme, as you see, this is a large molecule bind to the active site, right? So this enzyme is a catabolic enzyme, is in this example, right? So what will happen? That this enzyme, it will break down the substrate and it will become free by itself. Now, this is the enzyme, and let's suppose that this is the product that we get. Now, what is there? There was just one large molecule in the beginning, right? There was one large molecule of substrate. After the completion of the chemical reaction, we have got two smaller molecules. So one large molecule is broken down into two smaller molecules. We'll call this enzyme as a catabolic enzyme, a breaker enzyme, right? And if you observe, this active site is free. As the active site is free, so what did we learn? That this enzyme can be what? Reused. So the enzyme, as it can be reused, so what it means? It means that enzymes are required in small quantities. Remember this or not? Now, let's take the other example. Let's suppose that this is an enzyme and this is its active site, right? Again, there can be different types of substrates. Let's suppose that this is the substrate. This is the substrate, and these are the substrates. Now what will happen? That these two substrates, they can fit over here, right? So what will happen? That these two substrates, they will bind to the active site of the enzyme, and when they will bind to the active site of the enzyme, what will be formed? Enzyme substrate complex.
It is already broken.
Right?
It is already broken.
Yes, very good. Well done. Need to pay attention. If it is already broken, then what is going to happen next? Magic. Right? Now, these two substrates, they entered into the active site. Remember what is this? Substrates. In this enzyme, one substrate was added. In this enzyme, two substrates are added. Look at the magic. What will happen? That at the end of the reaction, this enzyme, it will become free, and as this is a builder enzyme, anabolic enzyme, what it will do? It will bind both of these substrates together to make a larger molecule. Two small molecules join together to make a larger molecule. Understand? So this is the example of an anabolic enzyme.
Sir, where are better enzymes in our body?
Everywhere. They are in your cells. In your nucleus they are present. In your cytoplasm they are present. Protein synthesis is happening or not?
Yes.
Amino acids are joined together to make polypeptides. Small molecules join to make large molecules. Anabolic enzymes. Alright? Now again, this is what? Active site. And it is what? Free. So it means that this enzyme will be what? Reused. Understand? So these are the two examples of lock and key model, which tells us that the shape of the substrate is an exact fit of the shape of the active site. Understood? Now, next model which we are going to talk about, that is the induced fit model. Now, in the induced fit model, it says that the active site shape resembles the shape of substrate. Now please pay attention. Being the exact shape and being resembling These are two not same. These are not same things. These are different things. Exact means perfect. Resembling means resembles. Not perfectly same. Right? And next point about this mode of action is that active site take the exact shape of substrate when substrate binds to active site. Now, if you guys remember, yesterday, just giving an overview, I gave you guys an example. I gave you guys an example of what? Gloves. Gloves, what do? They resemble the shape of a hand, right? But they are not exactly, let's suppose if I buy a pair of gloves, those gloves resemble the shape of my hand, but they are not the exact shape of my hand, right? Yes. But when I will put on those gloves, I will wear those gloves, they will take the exact same shape of my hands, right? So same concept is given over here. Now let's understand this with the help of an example. Let's suppose that over here, let's suppose this is what? This is an enzyme, right? And this over here is its active site. And the substrate for this active site is this. Now if you see that the shape of this substrate, this is what? This is substrate. The shape is resembling but not an exact match. But not an exact match. What will happen when this substrate and the enzyme, they will bind together? The enzyme will take the shape of its active site will take the shape of the substrate. Let me show you how. Now, what has happened? Look here. What has happened? That the active site is now exactly the shape of the substrate. And we will call this one as enzyme substrate complex. Understood everyone? Next step, what did we learn? That enzyme substrate complex is formed for a very short period of time. Right away what will happen? Now remember that this is a larger molecule I have shown you, right? So in this example I am showing you a catabolic enzyme, a breaker enzyme. Now what will happen? That this substrate will be broken down into two smaller molecules which will be called what? Products. And what will be released? Enzyme. Now interestingly, right away when the enzyme is released, the product is released, the enzyme comes back to its original shape. The enzyme comes back to its original shape, where the active site was a resembling match, not an exact match. Now the active site is what? Free. So as the active site is free, it means that enzyme can be what? Reused. Understood everyone? So when the enzyme performs its job, makes the product, and right away the product is released outside, what happens? That the active site becomes as it was in the earlier or before. Alright? So this is the induced fit model. The shape of the active site becomes the exact shape when the substrate fits into it. Now some enzymes, they follow this rule. Some enzymes, they follow this rule, lock and key model. But there are some enzymes which follow this model as well. The question is, how can we tell this? We can't see the enzyme working, right? We can tell this with the help of experiments which we perform. Understand? That these kind of models are performed by which kind of enzymes. Alright?